So it takes a lot of ATP to make this work. And we're moving on to metabolism inside of a skeletal muscle fiber. And I don't know what your background is, so at least in Bio 211, AMP1 at Norwalk Community College, we're going to introduce to you 3 major mechanisms that allow for ATP availability inside skeletal muscle tissue. The first of which is going to be transfer through substrate phosphorylation by creatine phosphate. The other is anaerobic cellular respiration. You know it as glycolysis. And then you actually have aerobic cellular respiration, which is glycolysis, Krebs cycle, and the electron transport chain that can be fed through not only glucose, but also fatty acids and amino acids. Okay, so in this next figure, you're going to see what I'm talking about. Up in the front, the top here, in part A, is ATP from creatine phosphate. And in times of plenty, meaning you're sitting on your ass right now, you're pretending to pay attention to what I'm saying, you have lots of ATP that can donate a phosphate to creatine. And creatine is a breakdown product for normal metabolism, and what it does is it stores it temporarily. And imagine now if you had to get up and sprint across the house to go to the bathroom in time or something like that. You could take that phosphate and reattach it to the ADP that had just been hydrolyzed by the myosin head. Now you have ATP, and you get to the bathroom in time, and you get to go to the bathroom without soiling yourself. Okay? Think about what I just said. We didn't have to go to the mitochondria. We didn't use any oxygen. This is when you are utilizing ATP that is in excess for the future when you may need it, when it's not in excess. Okay. The next is going to be if you have found yourself running to the bathroom and it's far, far away and it's up 3 or 4 flights of stairs and you're still trying to hold on to it. What's going to happen, of course, is you're going to burn through all of the ATP inside of your muscle cells really quickly, and then your body has to break down glucose that may or may not have been mobilized from glycogen inside either your liver or your skeletal muscle tissue. Sorry about that. And you break that down, you get 2 ATP molecules, but because you are running and you don't have enough oxygen coming in, that pyruvic acid is converted into lactic acid. And if you happen to make it to the toilet in time, then while you're sitting there doing your other things, okay, you're panting, you're how did I ever make it? That lactic acid can be converted back to pyruvic acid. And then that will travel over here to our mitochondria. So when you actually are doing all of that, what I mean by doing all of that is now you're breathing. We have the major mechanism for making the largest number of molecules of ATP. And up here you can see normal glycolysis breaks down glucose to give us pyruvic acid. That pyruvic acid will now travel into the mitochondria where we make ATP. When we don't have any glucose or glycogen anymore, you can use fatty acids that can be liberated from adipocytes, or you can use amino acids from protein breakdown. In both of these cases, or I should say all 3 of these cases, you need oxygen from hemoglobin to be transferred to myoglobin, although you could bypass it if you have plenty of it around, right? The myoglobin will have to store it a little bit longer inside your skeletal muscle tissue, okay, or your cardiac muscle tissue. And that's what's going to happen. So if we've made it to the mitochondria, we have to be using oxygen. So I've already explained to you what's going to happen here during anaerobic breakdown. But what I want you to do is I want you to pay attention to the fact that I'm showing you in great detail here that somehow glucose is used to generate ATP. It can be derived from blood as well as glycogen stores. In AMP2, you can learn where the glycogen actually, how we mobilize glycogen using glucagon. Okay? But then we're going to make, as a result of that, we're going to make ATP. And as long as oxygen is present, we go directly into the aerobic respiration pathway. If they're not, remember we make lactic acid. Inside your textbook, they would also call that fermentation. Okay? So aerobic is pretty straightforward, and what I mean by that is pyruvic acid enters into the mitochondria, okay, and is completely oxidized to give us ATP, carbon dioxide, water, and heat. Okay, remember the C6H12O6 plus O2 to give us CO2 plus H2O plus ATP, and now your body's warm. Gives you 36 molecules of ATP. Other parts of your textbook will say 36 to 38. Okay, whatever. Makes no difference, but it needs oxygen. Why do we care? Well, we care because muscles will fatigue. Okay? The inability of muscle to maintain force over time means that it has inadequate releases of calcium ion, or you've depleted all the creatine phosphate, or there's insufficient oxygen, or you've depleted all the glycogen, or you've added too much lactic acid. Okay? And all of these things will cause muscle fatigue, but that's okay, because afterwards you will pay off that oxygen debt, and in that paying off of the oxygen debt, you convert lactic acid into glycogen, and or lactic acid can be converted back into pyruvic acid. You can synthesize creatine phosphate and ATP when oxygen levels return to normal, and you can replace oxygen removed from myoglobin, meaning you've run out of it. That's actually how you pay off the oxygen debt. Your body actually works on debt, which is really both a good idea and a bad idea. But it's the only way it can work, to be quite honest with you. There's no way you actually can maintain excess of everything without taxing your physiology. Okay, so in the last part of our focus here, we want to think about, now that we actually have some energy, what do we do to actually control muscle tension? Because muscle regulation isn't an all or nothing proposition. Meaning if you wanted to pick up a pen, you don't exert the same amount of force that you would if you had to pick up a 50-pound weight. That's the way I want you to think about it, right? Tension or force varies, and it has to. It's dependent upon the rate at which the nerve impulse will arrive. Okay? It depends on the amount of stretch before contraction. It depends on the nutrient and oxygen availability, as well as the size of the motor unit, meaning how many muscle fibers are activated by one lower motor neuron. And you'll see what I mean by that. Okay, take a look here. Here is a neuromuscular junction by two somatic lower motor neurons. And notice how, in this case, this innervates multiple targets. So this would actually activate more muscle fiber contractions than this one here. Which one gives more precision? Obviously, the one in purple gives more precision, but the one in green may give you more force. So your body wants to figure out how to maximize that. To do this, what she does is she creates a twitch. And that twitch lasts from 20 to 200 milliseconds in length of time. But before the twitch actually happens, we have to release the action potential, which causes the calcium to be released. So during the latent period, after there's been electrical activity, there is the mechanical opening of those voltage-gated channels. That calcium binds to the troponin-tropomyosin complex, and now you form those cross bridges so you can actually get muscle contraction. But just because you have a twitch doesn't mean you actually have movement. In this cartoon, you can see the force of contraction, where we have our latent period, contraction occurring, and then you actually have to have a relaxation period. All these muscles have to take a break at some point in time, otherwise you exhaust them. Remember, fatigue. But we're going to sum them together to get something to move. Down here in blue is that action potential. Here are the actual skeletal muscle fibers summing so that you end up with, notice what I'm pointing, whoops, I didn't mean to do that, pointing out here, fused tetanus. And notice, look at the number of lower motor neuron action potentials activating the excitability. When you sum all of this together, that's what actually causes muscle tone. So a small amount of tension in a muscle is due to weak contractions. Large groups of muscle units are alternately active and inactive, so you can maintain some muscle tone, and in this case actually sustain it. You know what you feel like and look like after you've been to the gym for about 6 months and you've done concerted work. It's that muscle tone that keeps those skeletal muscles firm, as well as the involuntary muscle tone of, say, your posture to keep your head from slumping forward on your chest. When we put all this action together, we have two types of contractions. You either have an isotonic contraction, where the tension developed remains constant while the muscle changes in its length. Okay, so this is like picking up your textbook to open it and read it. And then you have isometric contractions, which is holding the book steady on an outstretched arm. And inside class, you remember, that's what we actually did. You can actually have concentric movement, where you actually have this from low to high. Eccentric is high to low. No movement is when you have the antagonistic muscles both working, so there's no movement at all. The last concept, okay, as we move into the final part of the chapter is, well, how does Mother Nature know how to make a dynamic moving organism? And she does this by varying the amount of myoglobin, okay, that can actually be used for efficient contraction. So you have red muscle fibers and you have white muscle fibers, and essentially it's not that they have no myoglobin, they have lower amounts of myoglobin. And red muscle fibers have a high myoglobin content. I was going to say, look at chicken legs versus, are you a chicken leg person or are you a chicken breast person, right? Myoglobin is high in content inside the legs. It has many more mitochondria and it has much more blood flow to it. Okay. And what Mother Nature does is she makes three types of fibers, okay, that vary in their ability to fatigue. Slow oxidative, oxidative glycolytic, and fast glycolytic fibers are what are used. Inside your body, you may have slow oxidative fibers. These are smallest in diameter. They're the least powerful of all muscle types, okay? But they generally appear to have more myoglobin. They make ATP for aerobic cellular respiration. They have a really slow twitch contraction phase. Don't worry about that. But look at this. They're very resistant to fatigue, okay? Individuals who are really good marathoners have a large amount of slow oxidative fibers, specifically inside their legs. Fast oxidative glycolytic fibers, or FOG fibers, are intermediate in their diameter. They have a dark red appearance. They can make a lot of ATP. They are moderately high in their resistance to fatigue. Okay? They can also generate some ATP by glycolysis, but primarily this is walking and sprinting activity. Okay? Think of Usain Bolt. Essentially all, I can't say all fast oxidative glycolytic fibers, but a good sprinter has many of these. Fast glycolytic fibers, okay? They appear white in color. They have generally few mitochondria, but one thing to keep in mind is that as the more mitochondria you need, cells can actually make them. They're adapted for intense aerobic movements for short duration. And I'm surprised they actually don't put sprinting in here as well, because essentially a sprint is a fast, intense anaerobic movement. Okay, weightlifting or throwing a ball would be one way to think about it. But here's the thing. It makes most of its ATP by glycolysis. Okay? Inside your textbook, there's a table that, of course, has all of these things for you. And farther you go along academically, you want to start actually making these tables yourself. See how these were actually put together. What are the characteristics? Okay, so slow oxidative, red. Fast oxidative glycolytic, reddish pink. Fast glycolytic, white. You can see there's the characteristics. You can learn how to keep them apart inside your mind's eye. Okay, and what Mother Nature does, of course, she decorates your body with different ratios of these. This is why I will never be a world-class sprinter. I don't have, the types of fibers that are required for that. Okay, but I'm also not a world-class long-distance runner. In fact, I'm not much of anything, but that's okay too. You'd sit back and watch them on television, although I do rather enjoy playing sports. So, obviously, everybody has different ratios of each of these. Okay, whoops. Let's think about how they're decorating your body, though. Postural muscles, pretty much high proportion SO. Okay, leg muscles have large numbers of both SO and FOG. Okay, so you can see what Mother Nature is doing here. By regulating the ratio of each of these individuals can change what they actually do really well. But let's get away from this stuff. It's as interesting as it is. It's more exercise science than it is anything I want you to think about. So in the last couple of minutes here, let's change the topic. Here's cardiac muscle tissue. And remember in chapter 4, we had these intercalated or intercalated discs that allow for action potentials to travel inside between cells. So the charge doesn't actually have to exit the cell. And that's going to allow for continuous rhythmic activity, major physiologic difference between cardiac and skeletal muscle cells. Skeletal muscle fibers require individual neuromuscular junctions to regulate their activity. Once you actually have a depolarization on a cardiac muscle fiber, you're going to learn in your future that it can pass throughout the entire section of the heart. The contractions last longer because there's less calcium available, and the mitochondria are large and numerous. That heart's been beating since before you were born. It doesn't stop until you die. And here's another interesting thing. It depends on aerobic respiration, okay? But what your textbook doesn't tell you, at least in this part, is yeah, sure, it can use lactic acid, but it can also use primarily fatty acids as the major energy source to make ATP. Smooth muscle, this is found inside the walls of your arteries, hollow organs, your lungs, hair follicles, pupil diameter, so on and so forth, right? And they're stimulated by neurotransmitter, hormone, and autorhythmic fibers, right? So we don't control them. That's the point. But they do have, some of them do have gap junctions, okay, similar to our cardiac muscle tissue. And what I want you to do is take a look at the fact that Mother Nature makes them generally different when you look at them. You don't see orderly sarcomeres. They are there, but they usually take something to activate them, and that's usually a dense body or a stretch that will activate them. So you actually literally have to stretch the muscle fiber to actually get it to move, okay? Or you send a signal to it. So there are two types, single unit and multi-unit, okay? And you can see here that the contraction between these two types, where you actually have sustained muscle contraction of low force level, is regulated by slowly letting calcium in from interstitial fluid from outside of the cell. That's what's important here. Okay, so by regulating the availability of calcium, you change the rate at which they contract. The action potential, of course, is coming from the autonomic nervous system. And remember, these are antagonistic. So you have both sympathetic and parasympathetic sending contradictory signals. You also have, in response to stretching, the ability to actually activate muscle control, meaning so when you actually eat a piece of pizza, it stretches your stomach, it starts smooth muscle contraction. And then hormones. The hormone primarily, remember, the sympathetic nervous system is going to be epinephrine. Also changes in pH, oxygen, and carbon dioxide, primarily pH and carbon dioxide, will cause smooth muscle to constrict as well. Okay? And this is what actually happens. So instead of actually having a sarcomere, right, you still have your thin and your thick filaments, but notice they're attached to these things that are called dense bodies. Okay? And what happens is once you get it to start, they still move. They shorten the distance between 2 points, but look what's actually happened to the cell. It's sort of like this web surrounding it now constricts and pulls it shorter. Okay? And now in this last slide, I want you to see that skeletal muscle itself has limited regenerating abilities. Cardiac muscle can undergo much more of it as a result of workload. By increasing its workload, you can actually change the activity of your heart. Okay? And likewise, the smooth muscle inside the uterus for females can retain their capacity for division. Okay? This is a muscular organ of the reproductive system that actually has to function over a long period of time. It has to be stretched. It has to replace those tissues, and that's one of the sort of odd, I won't say odd, required adaptations of muscular tissue. One last thing, because I got like 30 seconds here. I want you to, when you're reading inside your textbook, okay, to notice that instead of using actin and myosin here, notice that this is an intermediate filament that's actually holding all of these things together, but the actin and myosin in between here is still being pulled on. So it's really actually an interesting design for your skeletal muscle tissue, and the enzyme inside here is not the myosin that we actually used. It's a CAM kinase that actually is going to regulate this. So that's chapter 10. And we'll obviously focus on some of the mechanisms primarily to test you on. But, this is how you actually move. Because remember, it's all dependent upon you regulating. So here's a skeletal muscle fiber that actually has a series of fibrils inside of it that have Z disc to Z disc. And you regulate that by activating muscle contraction, because we can release calcium from a sarcoplasmic reticulum. You shut it off by stopping that signal. Okay. Have a great afternoon. Bye. Evening.